Hello, my name is Pinu Dial, and I'm a student conducting research under the University of Washington, Basel. So, antibiotic resistance is one of the greatest threats to global health worldwide, as it was predicted that by 2050, antibiotic resistance will kill more than 10 million people per year. So, because of this, a big concern is how does antibiotic resistance arise? So, one of the most major mechanisms is called horizontal gene transfer, which can happen when a plasmid containing the gene for antibiotic resistance conjugates with another bacterium to spread resistance. And this is often responsible for the hospital-acquired resistance cases that we see. So because of this, plasmids are critical targets in order to prevent the rapid spread of resistance plasmids in a population. But the problem is, plasmids don't contain any vital cellular processes, which is why it's been difficult for us to target them for antibiotic development. However, many resistance plasmids do contain a lethal system called toxin-antitoxin systems that have been nicknamed as suicide modules because of their role in causing cell death in the bacteria through an internal mechanism. So, because the lethal activity of the toxin-antitoxin system is mediated through protein-protein interactions, my research had the goal of using computational drug design to develop a small molecule inhibitor disrupting the interaction to artificially activate the system in bacteria containing resistance plasmids as a potential strategy for selectively targeting bacteria carrying resistance plasmids without harming other commensal bacteria. Okay, so the first piece was to identify the critical interaction sites between the toxin and the antitoxin protein in the lethal KIT-KISS toxin-antitoxin system found in resistance plasmid R1. And this involved calculating the interacting amino acid residues for contributions to binding energy using pocket query, which identified these residues as the most likely targets for the development of a small molecule inhibitor. So, given my target binding sites, I used the rational drug design approach called de novo design to link fragments together and optimize the molecule with a genetic algorithm with the following parameters for the scoring function. And the top scoring generation was selected as the final molecule for the binding site. And these were the molecules that were constructed with de novo design. Okay, so in order to evaluate for the efficacy of the inhibitors, a QSAR model was constructed to find a relationship between the molecular properties, known as the descriptors of a known data set of compounds, with the biological activity. So my model aimed to classify the compounds as either active or inactive, and this was constructed using the associative neural network method based on a known data set of activators of the Maziev toxin-antitoxin system, which has very high structural similarities with the KIT-KISS toxin-antitoxin system. So the model predicted 6 out of the 9 inhibitors as active compounds, and interestingly, 4 out of the 5 inhibitors that were targeted towards the top scoring interacting region were predicted as active. So along with current experimental data on active sites of the KIT toxin, this supports the hypothesis that this region from GLU66 to ARG72 is the most critical interaction site and the most promising target for the development of a small molecule inhibitor. So the second aspect that I evaluated my inhibitors on was the binding affinity. So using computational tools, the predicted values for both delta G and PKD of the binding of the inhibitor to the protein target were found to be energetically favorable and spontaneous. So using a two-tailed t-test, the p-values of the inhibitors binding to the protein target were found to be statistically significant in comparison to the control molecules. So in total, I used computational drug design to develop several inhibitors designed to bind to the interacting region between the toxin and the antitoxin in order to disrupt their protein-protein interaction, which frees the toxin from inhibition by the antitoxin and allows it to act lethally on the cellular target. So through computational predictions, 6 out of the 9 inhibitors were predicted as active compounds for activating the KIT-KISS toxin-antitoxin system, particularly the GLU66 to ARG72 region that consistently scored the highest in binding energy calculations between residues. And then some further theoretical implications that could be further explored may be the potential for selectively targeting bacteria containing resistance plasmids without harming commensal bacteria, since toxin-antitoxin systems are located in many low copy number resistance plasmids with no known homologs in eukaryotic cells. So thank you so much for listening to my presentation.